Okay, so in chapter 10, we're talking about photosynthesis. So we talked briefly about this at the beginning of chapter 9. We talked about the linkage between photosynthesis and cellular respiration. And so we'll revisit that idea today of this sort of global linkage between these two processes. And then we'll talk a lot about the actual process of photosynthesis. So photosynthesis involves reactions that convert energy from the sun to the chemical bond energy of ATP. And so, again, this is just that energy conversion. So this is the law of conservation of energy that we have talked about before, that first law of thermodynamics, which says that energy cannot be created or destroyed, but it is converted from one form to another. And that is exactly what photosynthesis is doing. It's capturing the heat energy from the sun and converting that to chemical energy. ATP is then going to be used to drive reactions that produce glucose. So the whole point of this process of photosynthesis is really to produce glucose, which is the food for the plant, as well as some other energy-rich compounds, which we'll talk about. But in order to do that, we have to have ATP, which drives production of that glucose. We know that ATP is a cell's energy currency. It's the way that your cells use and store energy, which we've already talked about in terms of cellular respiration. And so you'll see that again today as we go through photosynthesis. A lot of survival of organisms is dependent upon photosynthesis. In fact, even non-photosynthetic organisms depend on photosynthesis for survival because the main byproduct of photosynthesis, the molecule that is released by photosynthetic organisms, is oxygen. And we all, any aerobic organism who carries out cellular respiration and aerobic respiration needs oxygen to survive. So the first thing we have to talk about when we go through photosynthesis is we have to figure out how is it that we capture that energy from the sun. And we do that by using molecules that are called pigments. So a pigment is simply a molecule that absorbs wavelengths or colors of light. And every pigment has a specific absorption spectrum. And so we have to talk about and look at what these absorption spectrums are. It's basically a graph that demonstrates how much of each wavelength or color a pigment would absorb. So for example, if we have a graph that looks like this, I'm going to highlight the graph in green that we are sort of looking at. So it has very high levels of absorption in all areas except this blue region down here. So this is percent absorbance on the y-axis. So this shows you what percentage of light is absorbed by this pigment. You can see from purple and then through sort of green, yellow, orange, and red, we have almost 100% absorption for this particular pigment. But at blue wavelength, you can see the absorption dips, and we have very low levels of absorption. That means that blue light is reflected by this particular pigment, and whatever light is reflected, that is the light that you see with your eyes. So when we look at this particular pigment, what our eyes would actually see is the reflected blue light. All the other light from this pigment would be absorbed by the pigment. Okay, so the two main pigments that we are interested in when it comes to photosynthesis are chlorophyll A and chlorophyll B. So these are the pigments that are responsible for carrying out photosynthesis. And in chlorophyll A, we see blue and sort of violet lights absorbed the most. And in chlorophyll B, we see red lights absorbed the most. And so if we look at the absorption spectrum here, we have the solid line representing chlorophyll A and then the dashed line representing chlorophyll B. So if we look at A just for a second, we see that in this sort of purpley blue region, we have pretty high levels of absorption, up to about 60% maximum. When we get into the greens and the yellows, we start to see that absorption spectrum bottom out. So that means that these are the lights that are reflected by chlorophyll A. So this is the reflected light, or what we actually see. So a lot of photosynthetic organisms have very high levels of expression of chlorophyll A, and that is why the leaves that we see on a tree that are full of photosynthesis containing or carrying out organelles appear green, because they have very high levels of chlorophyll A that reflects that green light that our eyes see. Now chlorophyll B, you can see, so that's the dash line. It has sort of high levels of absorption. Again, here in this sort of bluish green area, we see pretty high levels, up to about 80%. And then we see another peak here in the orange range. But again, if we look at where chlorophyll B sort of bottoms out, it's here in this greenish yellowish region. And so that is why, again, the leaves that we see on photosynthetic trees that carry out photosynthesis appear green, because that is the light, the color of light that is reflected by those pigments. Now, there are also what we call accessory pigments. These are pigments that absorb light that chlorophyll A and chlorophyll B cannot. So here are the absorption spectrums for a couple of accessory pigments. You can see these guys actually have pretty high absorption levels in that blue-green, yellow region. But if you look over here, they bottom out over here in this orangey red region. And so these guys would certainly reflect these colors of light, the oranges and the reds and the yellows. And that is why when the leaves change color, there is a shift in the expression pattern of the pigments. We shift from having chlorophyll A and chlorophyll B being expressed to these accessory pigments being expressed. And when that happens, that's when we start to see the leaves turn to the oranges and the reds and the yellows in the fall, is because they are expressing a different pigment that's absorbing different colors of light. All right, so here's our overview equation for photosynthesis. We're going to start with water. This is H2O. So we know that plants can absorb water from the soil. We're going to also have some carbon dioxide. This is from atoms. And if we think about where carbon dioxide comes from, this is going to come, remember, from those air breathers. So this is one of the big byproducts of aerobic respiration. And so air breathers or aerobic respirators are giving off the carbon dioxide, and plants are going to use that carbon dioxide to carry out photosynthesis. So we have water from the soil, we have carbon dioxide from the atmosphere, and we're going to take that light energy from the sun. So we require that light energy. And what we are going to do is we're going to convert that energy into the high energy rich molecule glucose. So this is C6H12O6. So that's glucose. That's the food source for the plant. And then oxygen is the byproduct. So plants do not use oxygen. They think oxygen is just useless. And so they end up giving it off. And then we actually use that oxygen to carry out cellular respiration that we talked about back in chapter 9. So that is sort of the global linkage again between photosynthesis and cellular respiration. Now, photosynthesis takes place mostly in the leaves, sometimes in the stems of a plant, but most of photosynthesis happens in the leaves. So here we are, we're on the upper surface of that leaf. So we have a leaf, we've kind of zoomed in here on the upper surface. So this is the upper surface of the leaf here. And we have all these cells that are located along that upper surface of the leaf. We then have photosynthetic cells, so we can zoom in on these photosynthetic cells. And within the photosynthetic cells, they have chloroplasts. So that's these little green circles here are the chloroplasts. And so we can zoom in and sort of take a look at that chloroplast, and this is what they look like. So a chloroplast, just like the mitochondria, has a double membrane system. So they have an outer membrane as well as an inner membrane. And then they have these little stacks of membranes inside that sort of look like pancake stacks. These are called thylakoids. And so we're going to talk a lot about thylakoids as we go through the rest of this chapter. These are those little membrane stacks there. So this is where photosynthesis is actually taking place, is in the chloroplast of the photosynthetic cells that's located along the upper surface of the leaves. All right, so let's take a look at the parts of the chloroplast then a little more carefully. So again, I told you these little membrane stacks that look like stacks of pancakes, they're called thylakoids. They're located within that inner membrane of the mitochondria. They carry out what are called the light-dependent reactions. So we're going to divide photosynthesis into two parts, light-dependent reactions and light-independent reactions. So the light-dependent reactions take place here in the thylakoids. They are filled with hydrogen ions. So the thylakoids are filled with hydrogen ions, and that's going to be important for us. In just a moment, we'll talk more about that. The purpose of the light-dependent reactions that happen in the thylakoid is to take that light energy and convert it to chemical energy. So that light energy, remember, comes from the sun. The chemical energy is going to be in the form of ATP. So we produce three products here in the light-dependent reactions. We're going to produce ATP, we're going to produce NADPH, and we're going to produce some O2. NADPH is exactly like, it functions exactly like NADH that we talked about in cellular respiration. It is an electron and a hydrogen ion taxi. So remember, we talked about those back in chapter 9. The purpose for those is to deliver electrons and hydrogen ions where we need them to be in the cell. And so NADPH is going to do exactly the same thing that NADH did in cellular respiration. The other part of the chloroplast is going to be important is the stroma. So stroma. This is the interior sort of semi-fluid of the thylakoid, or I'm sorry, of the chloroplast. So it's almost like a toothpastey substance that is distributed throughout those thylakoid membranes. So I kind of say the thylakoid membranes are like the stacks of pancakes, and the stroma you can think of as like the syrup that sort of flows through those pancakes. It's a semi-fluid interior. This is where the light-independent reactions are going to take place. So again, this is sort of the second half or the synthesis part of photosynthesis. The point of these reactions is to convert CO2 to glucose. So we're going to start with the CO2 and we're going to end up with glucose in these reactions. So again, this is the food that the plant needs to survive. Here, we are going to use the ATP and the NADPH that we just made in the light-dependent reactions that take place in the thylakoid. So now we are going to use those two energy sources to drive production of glucose. All right, so let's talk a little bit about the light-dependent reactions. So again, these occur in the thylakoid membranes. We're going to use those pigments that we talked about, the chlorophyll A, the chlorophyll B, and the accessory pigments, to absorb photon energy. So this is light energy from the sun. We're going to use those pigments to absorb that light energy and drive the production of ATP. We are going to take a water molecule and split it apart. So we're going to talk about that. So when we take water and we split that apart, we're going to get hydrogen ions, we're going to get oxygen, and we're going to get some electrons. And so we'll talk about the importance of that. And then we're going to have an electron transport system just like we had in cellular respiration, where we took those electrons and kind of bumped them down the steps and sent them through the membranes. The same thing happens here in photosynthesis, is we get the transfer of those electrons and hydrogen ions through that electron transport system, and we're going to use the NADP+, converting that to NADPH as we do that. So we'll talk through these steps. All right, so the first thing that has to happen is we have to have these pigments that are going to absorb that light energy from the sun. So we use what are called photosystems, which are basically just clusters of the pigment molecules. So we take about 200 or 300 of those chlorophyll A or chlorophyll B or accessory pigment molecules, and we cluster them together. We group them together into what we call a photosystem. Again, these photosystems are located in the thylakoid membranes because that's where the light-dependent reactions happen. And their job is to absorb light energy in order to excite an electron. So the light energy is going to come in from the sun. We're going to absorb that light energy by the pigments that are located, make up these photosystems. And when that light energy hits those electrons, the electron gets excited and it starts bouncing around, sort of like a toddler that you have given 3 chocolate bars to, and they're going to bounce off the walls. And that's what that electron starts to do. It starts bouncing around that photosystem between the pigments that make up that photosystem because it's excited. It has absorbed all of that light energy from the sun. So as the electron starts to bounce around, so again, here's our photosystem. It's going to absorb that light energy. So we have all of our pigments, and again, there would be about 200 to 300 pigments that make up this photosystem. The electron is going to start bouncing around this thing. So it's going to bounce around like a toddler who has been given chocolate bars until it gets captured here in this particular area by what we call the photocenter. So the electron is going to be captured and stored, and we'll talk about that on the next slide. But that energy is passed around from one pigment molecule to another. So here we have our electron. Again, it's sort of bouncing around everywhere until it's going to get captured by the reaction center. Okay, so we're going to capture that electron in the reaction center. So energy, that electron flows randomly through the photosystem, or as energy flows through that photosystem until it is captured. This causes, what happens is when we activate that electron and excite that electron, it causes a water molecule to be split. So again, we're going to take that water molecule, we're going to split it into the hydrogen ion, the electron, and the molecular oxygen. So what happens is when that electron is released and it's passed down through that membrane system in the thylakoid, it activates the enzyme that catalyzes this reaction and the breakdown of water. So this electron is actually going to be stored again in the reaction center, so that when we absorb more light energy, we can excite that next electron and kind of start this process all over again. So it's like a circle. So this electron is going to get stored up here to be used later. Oxygen is going to be released into the atmosphere because plants do not need oxygen. They think it's a big waste of time. So oxygen is going to get released, and air breathers like you and I are going to use it to carry out cellular respiration. And then the hydrogen ions are going to be used to help drive the synthesis of ATP. So we're going to talk about that next. Okay, so here we are in the thylakoid, along the thylakoid membrane. So that's those little stacks of pancakes within the chloroplast. So here we are along the membrane. This is the inside of the thylakoid. This is the outside of the thylakoid. So here we have our water being split. So our electron just got excited. Here's our little excited electron because it has absorbed all that light energy from the sun. And so that has activated the enzyme that is going to split water. So we split water, we're going to have our oxygen and we have our hydrogen ions. So the oxygen, remember, gets released to the environment, so plants don't care about that. The electron is going to start bouncing down through this chain of membrane proteins, just like we kind of talked about in electron transfer phosphorylation in cellular respiration. You can sort of think about it as an electron sort of bouncing down some steps, or like a ball bouncing down steps. So that's exactly what it's doing here. As the electron flows through these proteins, flows down through this membrane, when it gets to the end or the bottom of the steps, there is this molecule NADP plus waiting for it. This is like the empty taxi. So it's like the taxi sitting there waiting to pick up a passenger. So when that electron gets there, that is the passenger for that taxi. And so when it joins or joins with NADP plus, we produce NADPH. So this is the occupied taxi. So this molecule now has taken on the electron and becomes the occupied taxi. Excuse me. So what happens is, as that electron is bouncing down that chain, it is pulling hydrogen ions inside the thylakoid. So you can see we're going to get hydrogen ions that are going to be shuttled inside this thylakoid. And it's going to set up this hydrogen ion gradient. It's going to be high on the inside and low on the outside. So this is very similar to the hydrogen ion gradient that we talked about in the mitochondria with cellular respiration, except the gradient is reversed. So now we have a high concentration inside the thylakoid, and we're going to have a low concentration outside the thylakoid. So again, that electron flowing through that membrane, flowing down that membrane system, is pulling those hydrogen ions in to the thylakoid membrane as it does that. So we're going to set up this gradient. So again, we have a high hydrogen ion concentration inside. We're going to have a low hydrogen ion concentration outside. And what happens is that is going to drive the production of ATP, just like it did in cellular respiration. So we've made our NADH, so that electron has joined with NAD+, and we've made NADH, and that electron has helped set up this gradient by pulling in all these hydrogen ions inside the thylakoid membrane. So now we're ready to make some ATP. So ATP synthase is located here on the membrane. And what happens is, as hydrogen ions flow down their concentration gradients, remember they're high inside, low outside, so they are going to flow out of that channel or that pump. And as they do that, they cause this reaction to take place. ADP plus a phosphate yields ATP. Okay, so this is how we are driving that synthesis of ATP. The hydrogen ions are flowing through this enzyme down their concentration gradient from an area of high to an area of low concentration. So hydrogen ion is powering this enzyme to catalyze this reaction to produce ATP. Okay, so now we just made NAD, so we just made our NADPH, and we made our ATP in the light-dependent reactions. We're now going to move on to the light-independent reactions where we're going to use those two molecules. So ATP is going to deliver the energy to drive the light-independent reactions. NADPH is going to deliver the hydrogen ions and the electrons. And carbon dioxide is going to provide the carbon and the oxygen. Now, where does the plant get the carbon dioxide from? It gets it from the atmosphere because we have breathed it out. So carbon dioxide is a byproduct for us and all other air breathers as we carry out cellular respiration. And so we have released carbon dioxide into the atmosphere, and that is where the plants are going to get that carbon dioxide that they need to produce the carbon and the oxygen. So our point here, the whole purpose of the light-independent reactions is to produce sugars in the form of glucose, which is the food source for the plant. Okay, so we do this, we carry out the light-dependent reactions through what we call the Calvin-Benson cycle. So again, this is a cyclical metabolic pathway that we use to carry out the synthesis part of the photosynthesis. So these are the light-independent reactions, again, which take place in the stroma, that sort of semi-fluid interior of the chloroplast. So in order to get this thing started off, we're going to take 6 CO2 molecules that the plant, again, gets from the atmosphere, and those CO2 molecules are going to join with a molecule called ribulose bisphosphate, or RUBP. So the number of dots here represent the number of carbons in these molecules. So CO2 has 1 carbon per molecule, but we have 6 molecules of CO2, so we technically have 6 carbons. And ribulose bisphosphate is a 5-carbon molecule. Okay, so again, I'm not going to ask you to memorize the names of the reactants in this reaction, but I do want you to understand what's happening in each step and what is being produced. So carbon dioxide is going to diffuse into the stroma of the chloroplast, again from the environment, and combines with ribulose bisphosphate to start this reaction off. So that reaction actually produces 12 molecules of a molecule called PGA. This is a very low energy molecule, so there is not a lot of energy potential here in PGA. It does not have a lot of chemical energy contained in its bonds. It's a low energy molecule. So we have to kind of pump it up a little bit and pump it full of energy, sort of get it beefed up. It has to go to the gym, it has to lift some weights, it has to get beefed up and ready to help us produce some glucose. So in order to pump some energy into PGA, we have to use ATP and NADPH. So these again are the molecules that we just made in the light-dependent reaction. So this is the ATP and the NADPH that we just made through in the thylakoids, the process of the light-dependent reactions. We're going to use these guys to pump up PGA that are then going to produce 12 molecules of what's called pGal. This is a very high energy molecule. So this is like your bodybuilder. It's beefed up, it's full of energy, it's been lifting weights, it's got big strong muscles, it's full of energy potential that we can use to make our glucose. And that's exactly what we do with that pGal. So we take 2 of those pGals, so each one has 3 carbons. We take 2 of them to produce 1 molecule of glucose. So throughout this process, we started with our 6 molecules of CO2. We have now made 1 molecule of glucose for our plant. So it's a 6-carbon molecule, so it's got those 6 balls that represent our 6 carbons. And this is what we call a phosphorylated glucose. It's got a phosphate group on the end of it. But this is the food source for our plant. So we made 12 of those very high energy pGal molecules. We just used 2 of them to make glucose. That leaves us with 10 molecules of pGal left over, which we are then going to use to regenerate our ribulose bisphosphate so that we can keep this cycle going and go through the cycle over and over and over again. In order to regenerate that ribulose bisphosphate, we have to break down some ATP. So we're going to use that remaining ATP that we made in the light-dependent reactions and convert that pGal back to ribulose bisphosphate so that this cycle can continue and we can keep making glucose for the plant. So that's what I expect you to know from the Calvin cycle. Again, I'm not going to ask you to memorize the structure. I wouldn't give you a blank page and say draw out the Calvin cycle, but I do want you to know and understand what happens in each step and how we get to this glucose production. All right, so there are a couple special types of organisms that we want to talk about here at the end of this chapter. These are called the photoautotrophs. These are organisms that get their energy from the sun. They get their carbon from carbon dioxide. They produce ATP, NADPH, glucose, and oxygen. So these are exactly the photosynthetic organisms that we have just talked about. These are your plants. Some bacteria carry out photosynthesis like this, and some protists carry out photosynthesis like this. But these are your very typical photosynthetic organisms that you think of when you think of carrying out photosynthesis. You can then have what are called chemoautotrophs. So these guys also carry out photosynthesis, but in a slightly different way. Instead of getting their energy from the sun, they get their energy from chemical reactions or chemical compounds. So dihydrogen sulfide, ammonia, iron, or other inorganic molecules. They still get their CO2, their carbon from CO2, just like plants and other traditional photosynthetic organisms. And basically, we're talking about there are some bacteria, a few bacteria, that carry out these types of reactions. But these are very different from your traditional photosynthetic plants that get their energy from the sun. Then lastly, we have what are called heteroautotrophs, and these guys get their energy as well as their carbon from feeding on other autotrophs or each other or organic waste. So there are some bacteria that carry this out, some protists. All your fungi are heteroautotrophs and animals are heteroautotrophs. So just in case you were wondering, yes, you as humans fit into this area. You do not produce your own food. You are not photosynthetic. You can't take energy from the sun and make food, but you obtain your food as well as your carbon by eating other autotrophs, by eating photosynthetic plants or eating other heteroautotrophs. And that's the way that we obtain our energy as well as our source of carbon. Okay, so that's the end of chapter 10. I will see you in class. We will cover, I'll see you in class for covering chapter 10. Thank you.